Hey friends, welcome back. Here we go. We're winding down our evolution unit. This is so exciting. We've we've covered a lot of information in a very short amount of time. So give yourselves a pat on the back. You guys have been keeping up very well with this. I only want to readdress a little bit of what we learned last week because I don't think it got enough attention. And the more I thought about it, the more it might show up, at least the concept of it. So I think all of you, after going through your assessments, you really nailed the difference between sympatric and allopatric speciation. I don't have to revisit those. You also understood very well the pre and post zygotic barriers. What I think I need to just spend a little more time on is how species can bifurcate, how they can speciate into two different directions over time. And that's really where this thing goes. So there's gradualism and there's punctuated equilibrium. Gradualism is as it sounds. There's this slow, steady rate over time. And as I mentioned in one of the classes, it's not even a measurement sometimes of the species as much as it tells you more about the environment. If you don't see a lot of change over time in species, that gives you a huge piece of evidence about how the environment was behaving at that time. Species change because the environment changes. Not a lot of species change, not a lot of environment change. And it gives us a window into the environment of that time. In a different way, when you see punctuated equilibrium, there must be some type of event here that caused the species to have to bottleneck really quick. So the environment changes really quick. You wipe out 90% of the ones who aren't adapted, the 10% that are left. They have just enough variation to survive. Those survival rates kick in. The variations begin with sexual reproduction, and then generation after generation, they reestablish themselves. And there can be several punctuated equilibrium events that would be an unstable environment over time. But you can also see that there's sort of a gradualism part built in where there is no change, right? It's really steady over time, and then boom, things change, and then steady over time, and then boom, things change. Punctuated equilibrium. So there's the two differences in them. This gets to the point of what are the conditions that really favor species evolving? And we want an unstable environment. I say if we want evolution to occur, we want an unstable environment, but we also want to see a lot of variation within the species. Why? It really favors, first of all, those individuals with variations. And second of all, those variations are favored in an environment that's constantly changing. So on this graph, the stability of the environment can be thought of as either unstable, and we want to make that the highest level, or really stable. So give you an example. The bottom of the ocean, 5 miles down, is wicked stable. It doesn't change. It doesn't even have weather, man. And if it does, it has like micro weather. It might go up or down by a tenth or 100th of a degree, but that's about it, right? So species down there have remained unchanged for millions of years. We have living fossils of species that are alive right now because they have been in existence the way they are for so long, the coelacanth being one of them. Unstable would be pretty much anywhere up here terrestrially. Mountain ranges, grasslands become floodplains. Floodplains become deep lakes or rivers or inland seas, and then they dry up, and then they become arid, and then there's like all of these shifts that land can go through. So there, and of course, there's a varying level of that, and that has to do with the latitude that you go on the Earth. Also, if you have just asexually reproducing organisms, there really isn't that much variation between them. So they are not going to be favored for in a really rapidly changing environment because, for obvious reasons, there aren't enough variations among them. And the ones that are successful would reproduce, but then you only get that one variation. It doesn't keep up very well with sexual reproduction. But if you have a lot of sexually reproducing species or individuals in that population, and there's also a lot of mutation, you get a higher mutation rate, boom, dude, you hit this real big sweet spot for evolution to occur. Now, in an unstable environment where you also have low variation, you hit a different type of sweet spot. And this is the extinction sweet spot. That's really how it happens. In a different way, and it's important to say it here, as species evolve, right, and they become more adapted to their environment, and as the environment becomes a little nichier or a little nichier, right, you really do start to see that cause extinction as well. Let me sort of explain why. You get a very basic, rugged species like bacteria, or sort of a generalist like a squirrel. They can adapt pretty quick. There's enough variation in them. They have a wide range of food. They can habitat in just about anything. But then as you start to get a little more specific, like humans, we lose our fur. We have a pretty high metabolism. We aren't very good claws, teeth. We need sharp things to kill each other, or our food. I didn't mean to say that way, but it's true about humans. The more we become adapted to little environments that we live in, if that environment suddenly changes, we are so adapted to the environment that we've become adapted to, that sudden change can cause a rapid extinction. The woolly mammoth is a fabulous example. It wasn't really made for, its size is what made it really good at thwarting off its predators. It was a herbivore, and then as two things worked against it, super fast climate change came out of the Ice Age, and humans, they just couldn't keep up with it. They had become so perfectly adapted at what was happening around them that when that sudden change happened, it was over. It was over for the woolly mammoth. That's what I mean by specificity can cause extinction, just like low variation can cause it. So, all right, moving on. This will be a nice transition into this week's lesson. This week is all about evidence of evolution. What evidence exists to demonstrate the species have been changing over time and will continue to change over time? Enter Richard Lenski. In 1988, Richard Lenski took some E. coli, and it was a really well thought out experiment. He selectively bred those E. coli to be genetically identical to each other. Just kept breeding them, breeding them. And the way you would do it is you take a petri dish. This is not a petri dish. This is my phone. And you create what's called a lawn of bacteria. So I take my E. coli and I spread it onto the petri dish with agar, and then I smear it all over there and I put it into the incubator, and I come back a day and a half later. And what I find is that I have a beautiful lawn. It's completely covered in bacteria. When they show up in these little colonies, these little bubbles, you can scoop a colony out, transfer it to a different agar plate, lawn it out again, and keep this going until you've removed all of the extra bacteria that were different from each other. You can keep going, and every little bubble, every little bump that shows up is one colony from one bacteria. Now, sometimes those colonies will merge, as in the first lawns that you made, but after you get about 50 lawns going, you get 50 deep, you're going to have genetically identical bacteria. Okay, so that was a really big setup for what he did. Creates them, and I think there was a dozen. I don't know. You can count them for yourself. I think there were 12 individual separations. So he takes that lawn and he puts them in 12 different isolated petri dishes and plates, and that represents the allopatric speciation. They were same conditions, right? He's going to incubate them at the same temperatures. He was going to do everything the same, but now they are barriered from each other. They can't mix genes. Sometimes bacteria don't sexually reproduce, but what they do, I said do, what they do is they can extend a little pili out. They can extend a little pili out, and they can send over some of their plasmids and exchange plasmids that way. Every time a bacteria dies, some of its genes spill into the environment, and other bacteria will pick it up. And in a population where there's a lot of different bacteria, that's going to matter. If they're all the same, it shouldn't matter because you're just picking up the same information you had a minute ago. Okay, 12 identical colonies are going to undergo 12 identical worlds, right? Nothing's going to change. So nothing should change unless evolution is real and one of 5 of the Hardy-Weinberg conditions are going to be broken, right? So there's not going to be natural selection. They don't mate, but they have their ability to die and share their genes is not limited. There isn't going to be any gene flow, between them, right? So we've cut off gene flow, but also we can't account for mutations and huge size. I mean, we're talking about bacteria. There's enormous, millions and millions in each petri dish. So you've really cut off their gene flow between the petri dishes, and you're not going to control mutations, but you're going to allow them to happen. Watch what happens. It does take a little bit. 25 years. And in the whole evolution scheme, it's a microsecond. But after 25 years, they changed. The size of them changed. The shape of them changed from petri dish to petri dish. And one of them evolved the ability to use citrate as a food. Now, you guys have already done Krebs cycle. You know that after glycolysis, you dump the pyruvate into the mitochondria, and then you have your coenzyme A, and it dumps off one of the carbon dioxide. So you have this coenzyme A, CoA, that goes in, and then it goes through the citric acid cycle, and you lose the rest of the carbon dioxide, and you generate a little bit of substrate level ATP. But that citrate was able to be metabolized further for food. That is pretty huge, right? That's like saying, like rabbits. Sam McRae, you've got a rabbit, man. Your rabbit eats its own poop, and then for one turn of it, that's like that. Like your waste is going to give you a little more food. That is super important, and that's just a little piece of evolution inside bacteria in this one spot. That says, what was this? 40,000 generations. He's up to, my God, I think he's past 100,000 generations now, and there are further. Look him up on the internet. It's a fabulously well-designed experiment. It's one of those where you're like, man, I wish I had thought of that. Or, that's a really good one. I wish I had been involved. But yeah, that represents, that little 25 years and 40,000 generations represents almost 1 million years in human evolution. It's pretty cool, man. Good stuff. Good stuff. This goes back to natural selection when we covered Harvard right in the beginning of this unit, where there was, in the beginning, there was no, oh, I got my pen. Why don't I just use that? There was no antibiotic, 1 times, 10 times, 100 times, and then in the middle was one, that's black, so I'm going to have to do an arrow, 1,000 times antibiotic in the middle. And I think they said it took 11 days for the bacteria to mutate enough to evolve to a state where they could withstand 1,000 times the antibiotic. Good stuff, man. Good stuff, good stuff. Okay, let's get to the evidence. The evidence for evolution comes in a few different, before I get to the proficiency scale, it comes in a few different pieces, the 5 that we have to remember: fossil record, embryology, homologous structures, biochemical homology or biochemical similarities, and then, of course, biogeography, which is my favorite. So let's go back to the scale and talk about that for a second. Let's talk about how to read the scale. Our targets are over here, right? This is our essential learning question, a less essential learning target. Evolution is supported by evidence on a cellular, molecular level, also a structural level. And you should be able, to the end of this, be able to explain how evolution is an ongoing process and how it is supported by the evidence of evolution. I haven't yet, and I'm not going to make a promise that I will, but I'm going to try to get you a Quizlet for all of these terms. If I don't, you really have to. These are pretty important terms. Make the effort. By the end of the week, everybody is going to be at least here. This is the base I need every one of you at. So when it comes to evidence indicates common ancestry for membrane-bound organelles, they're talking about the mitochondria. Like the mitochondria is a bacteria that has just evolved to be symbiotic with our own cells. The idea of linear chromosomes versus circular chromosomes, and then the idea of introns. We didn't cover viruses. Introns sort of go along with the idea of proviruses. This is all from the AP. This is all from the AP standards that they lay out for you. I would be most concerned with that one. Again, there's good to know and there's need to know. In a normal year, those bottom 2 would be good to knows. The A is a need to know this year, and that's where we want to be. Okay, endosymbiont theory. All species evolve and continue to evolve, so our genomes change over time. We've got the fossil record. just the evidence that bacteria can become resistant to an antibiotic is a micro evidence of evolution. And then pathogens, of course, evolve over time. So what I want you to do to be, if you really want that 10 at the end of the week on your assessment, if you want to get those 4.0s that so many of you continue to get and work so hard for, you got to be able to evaluate. I got to give you an example with some evidence in there and say, is this good evidence or is this bad evidence? And if it is good evidence, why is it good evidence? Support your answer. And if it's bad evidence, you have to be able to tell me why too. As always, I try to leave sample questions or sample examples at the bottom of what to expect and what those things would look like. So we're going to revisit this at the end and see how we did. Scientific method, you observe something, you make a hypothesis, you run an experiment, you gather some data, and then you make a conclusion, yes or no, I accept or reject my hypothesis. You do that enough times, you should be able to make a prediction. That's the way science works. I should be able to use the data that I've collected to, in a unique situation, apply that and have similar results, or at least tell you what should happen down the road. This evidence that we're gathering should make predictions about, one, is evolution going to continue to happen? Two, if I haven't been able to see where it happened, when I do see it, I should be able to predict how it's happening. Those are predictions. And I hope you're going to be pleasantly surprised to see that it does work. So those are the 5, right? We've already talked about the 5. Let's start with the fossil record, man. Going back to Hutton, going back to Lyell, going back to Smith, all of those scientists who were involved in getting us the geological time, deep time, and the idea of the fossil record and how important it is. That's where it started, but it hasn't gone away. I remember when I was a kid, and I don't think I'm unique among all of us here, that fossils always fascinated me. The idea that I'm holding something in my hand, and as like a little 8-year-old, you don't get 200 million years in your head, but you do understand old. You do understand before you. And this thing was here way before me, and it lived in a world that looks nothing like where I live right now, and yet it's here right now. That's cool. But what I didn't understand was I thought everything fossilized, right? I thought, okay, we can just go back and gather everything. This should be just a record. And what I now understand is kind of like the box my mom gave me a few weeks ago from when I was a kid of some stuff that I had. And if you were like, hey, Mainville, what were you like as a kid? And I gave you this box, it would be a pretty poor representation of what my childhood was like. There might be a toy in there that was important to me, but there's also some weird projects that I did as a kid, or there's like a ribbon that we have absolutely no idea what it went to. I don't know if I won the boxcar derby, a race, or somebody just felt bad for me and gave me a ribbon. Like I can't tell what it was for, so I might have to do a little more research on that. That's sort of the way the fossil record is, man. Fossils are really hard to lay down. The conditions for a fossil are near impossible, yet we have them. And given enough species have lived on this earth versus the amount of fossils that we don't have the complete picture of what used to be here, but we have enough to fill in the majority of the big gaps, right? In the test that we took or a practice question that we took last week, 130 genre of mammals have evolved, right? That's not the species, right? So the gray fox, the red fox, there's one more fox, I can't remember who he is, but that's like in the genre of foxes, there's probably 6 or 7 species, right? And that wasn't a bark. So 130, you blow that out to be, I don't know, 400, 500, 600, 700 different species within there. Not everything's going to be fossilized. Millions and millions of years from now, we may have fossils of a couple of the elephants that were around, and not even the different species. There would be like, some of those are going to be lost. Maybe one of the primates, maybe, I mean, obviously us, there's enough of us floating around, but there's not going to be a perfect record of the snapshot that exists right now. And nor should we expect it to be, because fossils are hard to make. I think I spent too much time on that, but it's a really big point to make. So uniformitarianism, the Earth has been undergoing the same conditions over time, which is also interesting because we've lost fossils because of subduction, because of geological plates moving around, because of erosion. Once they get up, they get eroded away. But it does give us a window, and it does give us enough evidence to know that the species that are here today came from distinctly different species that came before us. Over here on the left-hand side, this is horse foot evolution. So these are millions and millions of years old, but you can see that that middle toe was probably selected for. It must have given it some sort of sturdy balance because that toe kept getting selected for until it becomes a hoof, right? Okay. I don't know that you're impressed by that, but it does show we have the fossil record and the skeletons to go along with it. And then you see that early on horse evolution, you almost have like an insectivore set of teeth, but you do, it's called a diastema, the gap between the front teeth and the back. You still have a big gap in that herbivore gap, and you start to move towards what we call the ungulates. You have that grinding back tooth, you have that big gap in the nipping front, and there is a steady progression. You don't always get what they call the gap, not the gap, the missing link in that gap, because, I mean, this is a missing link of these two. But you can sort of follow that to a fool's end because then you expect, well, where is it for here? Well, if I do find that, then you can make the argument for where's the one here and where's the one here. And if you make that argument, then where's the one here and where's the one here and where's the one here. You can keep stretching it out. And as I just said, these things are hard to make. So the fact that we have them is beautiful. You can't expect to fill in all of those gaps in that study. You gotta be a little, you have to be a little comfortable with the gaps that you do have and be able to fill them in with the fossils that are left. Example of Archaeopteryx is a great one. Archaeopteryx has breastbones like a lizard. It has neck bones like a lizard. It has a beak like a bird. It has front claws like a lizard, but it has feathers like a bird. It has a tail like a lizard, but it has hip bones like a bird. Like there's all these hybridizations occurring. And this, when it was found, we're like, hey, this is the missing link between all of the velociraptors and all of the birds that we have. And we now know, and it was foolish to say that, Archaeopteryx actually belongs way over here. It is not the missing link, but it is a branch on the tree. The evolution of birds follows a much different path, but it's pretty cool that the idea of the feather is not just a one and done. There's lots of transition fossils. There's lots of families within the bird family to show that flight was actually going in a bunch of different directions because of feathers. Okay. A very, 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 very strong piece of evidence for fossils is that you always find them in order. And that should allow you to make predictions. We've seen this before. And this actually gives me chills every time I say it because it's such a beautiful story about how well this type of evidence can make a prediction. Enter Neil Shubin. Neil Shubin was what's the word I'm looking for? He's a ichthyologist, which is for fish. I know Jake didn't like the word icht in class. He's an ichthyologist. He loves fish. And he wrote a book called Your Inner Fish, and I highly recommend reading it. It's fascinating. But then he also took a job in charge of an anatomy and physiology department. So he goes into the human aspect, but he's really good at fish, and he's blown away at how much similarity there is between us. I'm not going to give the book away. Read it. But in Neil's spare time, because this is just what Neil does, he was very interested in how do you find the jump from water to land in species. We know the evolution of fish is first when we talk about chordates, right? Our chordates. So the fish evolve and we have ichthyosaurs. We've got all these really cool things in the ocean. But then at some point, you've got a mountain lion. Like where along the geological time span do we point and say, this is where you used to have fish, and this is now where you have land animals, and this is what we're going to focus on. Right? There has to be that point. So what do? You gather your evidence, you find out what you know, and then you start to make predictions. And so in the fossil record, we know that the jawless fish are about 375, 380 million years ago. That's where they are. But we also know that about between 365 and 370 million years ago, that's where we start to see our first land animals. Let's make a prediction. We should be able to find a fossil at roughly 370 million years ago that will be the transitional fossil. you shouldn't find, if you don't have any land animals, let me find my cursor again. If you don't find any land animals before this, then you shouldn't find any land animals before that. I said it the right way. If that's where they start, you shouldn't find them before it. And you don't. You really don't. I'm not just saying you don't because we haven't looked. You don't. Do you find them afterward? In spades. You find them everywhere. So that is the sweet spot that we're looking for. Okay, that's one thing. The second thing is fish just don't flop out of the water and start having fun and eating insects and whatever else a fish hybrid would do. You sort of have to have a condition. It wouldn't be up in like, I don't know, British Columbia or Newfoundland or anything where they have like the fjords, right? You wouldn't swim in and then start hiking your way up a steep cliff. You really have to have a certain condition. You have to have tidal flow. And I mean, if we work through it, this is what you would expect. You'd want an animal that's a sea-dwelling animal to be able to get up to a shallower area, and the water would probably recede. You'd want to have food there for it, and then the water would have to come back because, of course, it's still a sort of a fish. And so you would want an estuary-style area. We could spend more time talking about this later. I'm trying to, for brevity of notes. So Neil digs into his old geological textbooks, and he looks at where would estuaries exist at about 370 million years ago. And as it turns out, at the top of Canada, there is a certain layer of strata that was an estuary at about that time. So if we're going to start looking, and I'm not just saying that this is the source of everything, this is a good spot to look. This is one of the spots available. At 370 million years ago, this was an estuary. They'll start digging down. Let's find some exposed strata that dates to that area and see what we find. First time, fail miserably. Snowstorms. Dude, he goes up in July, and it's like between 0 and 20 degrees the whole entire time. It's one of the most muddy, miserable, cold things he's ever experienced. Second time, fail. Third time, fail. Fourth time, fail. This is a really good exercise in scientific experience. Fail, 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 fail. And his sixth time, six summers, 6 years of your life dedicated to doing this, he finds what he's looking for. Not only did he find what he was looking for, he found it in the strata level that he said he would find it in. And it actually looked almost identical to what he said he was going to find. So the perch that we catch, the fish we catch around here, have what they call a ray fin. It has like a lot of little bones. The lungfish has a different fin. It has this really long, almost like a tail-like we saw in Archaeopteryx with these tiny little bones that come off the side. And then you have what are called the rhabdicians. And the rhabdicians actually have a primitive humerus, the two-bone ulnar radius, and what you would call phalanges and metacarpals. This is a fish. This is a tetrapod. This type of fish actually has what we're looking for. And tetrapods, of course, like us, we've got those, well, we're bipedal, but you get what I'm saying with the chordate part. We have a humerus, we have ulnar radius, we have our carpal, metacarpal phalanges. And he finds Tiktaalik. Tiktaalik was named for the Inuit tribe that were there. It had the conical, not conical, but the flattened conical shape. It had the bone arrangement that it would find. You would find it was sort of the tweener between early tetrapod and late fish. Exactly as he said he would find it in the strata that he said he would find it. It makes the most sense. So cool. So cool. It made the prediction. Okay, so here's where Tiktaalik is. And Tiktaalik, Tiktaalik is really now like our ancestor 370 million years ago. That's how far back legged animals go. And Neil Shubin is forever going to be remembered as the one who found, not just found, it wasn't like he was walking along in Canada and fell down and go, hey, look, there's Tiktaalik. He used the scientific method. He used the evidence of evolution to predict precisely where it should be, and he found it. That's the power of this evidence. There's your living fossils. If you're a fisherman in the area, we do have living fossils. There are fossils of bowfin. Bowfin are an ancient species that we have fossils of, but yet they exist today. Pretty remarkable. On your left, that's the coelacanth that I was describing earlier. All right, enough about fossils. What do we know about fossils? They lay down an order. They always lay down in a predictable order. Species are always found in that order. And you can know something about the environment too, where the levels are that you find those fossils. Embryological evidence, this sometimes gets lumped in with the homology stuff because the similarities, but it's important for us to separate it for today. Ernest Haeckel had a man crush on Darwin, was really into the idea of evolution. And I preface this with, don't ever lie, don't ever make something up, don't ever embellish anything, especially in science. Ernest Haeckel was an embryologist, and he started following Darwin's stuff, made total sense to him, and then wanted to find evidence to support it. And that's the biggest mistake you can do with anything. You don't ever want to find evidence to support something. If anything, you want to find evidence against it. And if you can't find that evidence, the absence of that evidence actually supports what you're looking for. So what he should have been doing is just looking at embryos and seeing if the similarities do support selection. These are his drawings that he made. And let me explain what you're seeing. So here we have fish. And then we know fish evolved into salamanders. Those are our first tetrapods. And then we get the reptiles. And then we have the aves, right? We saw the velociraptor breaks off into one area for the birds. And then down the road, you have this series of mammals. What Haeckel saw was it seemed that species go through very similar embryological stages. Similar in that if you're a fish, you go through a fish stage. If you're a salamander, you go through a fish stage, but then you go through a salamander stage. And if you're a reptile or if you're a bird, let's start with reptile, you go through the initial fish stage. Your embryo looks like a fish embryo. And then you sort of go toward a salamander embryo. And I'm not saying you develop into a salamander, but there are stages within the salamander embryo stages that the reptile resembles. And then it goes into the reptile. And this is true for mammals too. We actually go through a very quick fish stage, and then it's really quick, salamander stage, and then for a little while, we do spend some time looking like everybody else's embryo. We have a tail. We have these pharyngeal gill slits, these pharyngeal arches that in some species become gills. If you're a shark, boop, 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 you got your gills. As you start moving away from fish towards us, some of these become bones, some of these become ears. They develop into, I have a diagram for it in a minute, but for us, it's our jawbone, it's our hyoid, and it's part of our ear bones that these cartilaginous gills used to become in those fish are now in us. Well, the drawings are on the top and the actuals are on the bottom. And he did embellish his drawings to make it look a little more like what he wanted it to look like. And to this day, right, that was 1875. To this day, it still gets up at like, look at how horrible the evidence for evolution is. That's a 150-year-old argument, dude. There's a lot better evidence nowadays, and I get where you're coming from. Yeah, shame on him, but you're hyper-focusing on the wrong target. If that was the only evidence and how bad his drawings were, we wouldn't be here having this discussion. There's mountains of other evidence. My lesson for you is don't embellish. Say what you have to say. Tell the truth always. Even if you take away the embellishment of his drawings and you look at the ontogeny, ontogeny is the embryological development, you do see the same exact phylogenetic tree play out. We do go through all those stages. This isn't an embryo course, so I think I have a drawing. Yeah, I could show you stuff and you would have no idea what you're looking at unless you were in an embryo class. But something really important to show is that we all have tails. If we don't have tails, why do we have tails as an embryo, right? Why do we have tails as an embryo? I'll ask again, why do we have tails as an embryo? We don't have tails now. Other species that do have tails as an embryo and then have tails. Why don't we keep our tails? It's because we go through that stage, apoptosis takes over, and then it boogies back. People are born sometimes with tails, by the way. It's interesting. Pharyngeal pouches, pharyngeal arches, those end up becoming, they're right here, and in this embryo, they're right there. Those end up becoming different things in different species. For us, each arches, as I said, become different types of cartilage and different types of bones for us. They're all controlled genetically, and mutations, you can see that, let's see here. Some of it is conserved, some of it is mutated. So if you look at a shark and you look at its gill arches and how they develop into the gills, that's pretty important. Even the fish, zebrafish, has a little bit of a differentiation, but once you start getting up to mammals, those gill arch genes develop into different things like your hyoid bone and your mandibular cartilage and different things of that nature. There's been mutations along the way. But it's still a homology because there are similarities as we go. Homology, I don't know if I said it yet, homology, as we already know, means similarity. So there's similarities between species over time. All right, now let's get into true blue, what you would have called beginner biohomologous structures. You remember, yeah, I compare a human bone, human arm against a chimpanzee arm. They're almost identical. They have longer fingers and their thumbs down a little farther. But that's to say that we have the humerus long bone, we have a two bone, we have a multi-bone carpal, and then we have the metacarpal and phalanges. But then there's variations of that along the way. If you take apart a whale's arm, whales are mammals, man, they're not fish. It has a humerus, it has an ulnar radius, they have carpals and they have phalanges. Those phalanges are like bat phalanges that have stretched out and get covered with skin and make wicked awesome flippers. So yeah, man, it's there. Okay. And that's true across all of the mammals that you can do it. But then when you get outside the aves, they look a little different. Their little tiny little fingers arrange themselves in a different way. And the wing structure is a little different. And you'd expect that because the farther you get away from being common with something, the more you expect there to be differences. And you find that species who are closely related have much greater homologous structures in common. Species who are farther apart have fewer homologous structures in common. Yeah. We know that. Okay, so this is a good one. DNA, if you can get it, is the strongest evidence because morphology may look similar, right? The flipper of a whale and the fin of a shark look remarkably similar if you don't know what you're looking for. But the structure of it can be really different when you get underneath the skin, and the genes that control it, forget about it. They're not even close. So that biochemical stuff is really it's the DNA, man. It's the coding that goes into it to find out how similar or different they are. I'm going to skip over this one because I'm trying to get to the good one here. Let's compare 3 types of blood: chimp blood, human blood, and rhesus monkey blood. And just as an aside, you're familiar with A, B, and AB blood, I'm sure by now. But you probably also know that there's an A positive and a B positive, or they say like B negative. This positive and negative refers to an extra protein besides the A and B protein. And it's called an Rh factor. Why? God, science is so weird. Why? It's because it was discovered in rhesus monkeys. And if you look at the first couple letters of rhesus, you could not help but notice that it's an Rh. So that's why we have it. We discovered the protein in rhesus monkeys. We do have it. Weird, right? We're all primates. Anyway, glutamic acid. If you're not familiar with glutamic acid, it's an amino acid, and there are 2 codons that code for it, GAA and GAG. And when you assemble a protein, it doesn't matter what order those go in. If you've got 20 GAAs in a row, you've got 20 glutamic acids. If you've got 20 GAGs in a row, you still have glutamic acid. Because of the wobble, these are both purines, you're going to get the same amino acid. So it really should not matter which one's where. Here's a codon chart to represent what I'm talking about. When you look at the hemoglobin that they code for, and you compare the chimp and the human and the rhesus monkey, there's a stunning fact that comes to light. I'm going to change the white because I have to write on black. 24, 28, 31, and 117 refer to the amino acid position. The hemoglobin is a pretty long protein. So at the 24th amino acid, there is a glutamic acid. And the glutamic acid in humans is coded for by GAG. And in chimpanzees, it's both GAG, and in rhesus monkeys, GAG. It could be a GAA, but it's not. Okay, Mainville, that's one. Boom. At 3 more positions, 4 more positions, at a total of 4 more positions, you find GAG. Okay. But where you find the GAAs, right, in the 122nd position, you find at the 23rd position, they are homologous. That shows we have very strict homology with the rhesus monkey and chimp. Our blood types are very, 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 very similar. This goes to the homology of the hemoglobin molecule. Again, that's one example. I know, man, but there's like 25,000 genes, and you find this as like 98% with, you take humans and you take chimps, and this is the kind of spelling accuracy that you find. We are that closely related where you could have you could have spellings. Where are you? Right here. You could have spellings for UCU, UCC, UCA, and UCG for a serine, and where we have it, we have identical ones for chimps. And 1.5% of the time, it's different. That's the homologous structure of biochemical similarities. Okay. Let's get to the last one, biogeography. By the way, how do we use biochemical similarities to prove that there's evolution? We do it a couple different ways. You can look at species, and you can take blood work, and you can find their common ancestor, and like at least predict their common ancestor. For some fossils, and I use that term loosely, you can find species that are between 1.5 and 50 million years old. You still have a chance to find some DNA. You can test that to find out how much homology you have in your biochemical structures. But it has really opened the door to the phylogenetic trees. And there has been some rearranging because of morphological similarities. You look similar, but man, once you get that DNA, you're like, yeah, that foot isn't really that similar to the other foot now that we consider it with the DNA evidence. And we've been able to find how similar we are to other species and how similar species are to each other. That's nice for knowing. What if you have a species who offers you some, I don't know, cancer curing properties, disease curing properties? If you know its genes and you know that it's related to the other species through common ancestors, you might be able to find other species that can generate that same cure. There's a lot of applications for this. If we had more time, I'd go in deeper. Let's get to the last one, biogeography, the living land, right? Where species live is just as good as evidence as where they don't live. This is our distribution through an artist's rendition of animals across the world. And I'm going to ask you, honestly, take a pause and just look. I'll wait. All right, what you should notice is there are some unique animals to different continents. There's some crossover, I guess, the alligator and the crocodile, okay, and one over here. And the camel seems to have some latitude on this way, and I see some squirrels over here, and I know I have squirrels that are not represented over here. Of course, birds can move around, but we have a different kind of wolf over here than we have over here, but there used to be a land bridge, so I get it. But there's also some unique stuff, man, like giraffes. Boom. Not on the other side, in spite of having the same latitude. Different types of little primates on this side that we have over here. Is that an aardvark? Looks like it. Different cat animals, parrots. Australia, before it was invaded by humans that brought placental mammals, it was exclusively marsupials, little pouch animals. So where you find species is sort of like an adaptive radiation idea. They got there, and then they blossomed, and that's what you have. That also says if you go back, right? Like, how is it? I don't see crocodiles swimming across the ocean. It also lets us know that when we go back in our geography, we know that we had Pangea, right? And there was some opportunity for species to cross over to other land masses. And then once those broke away, there's our allopatric speciation event. Then we can start to divide them up into different species. Modern species, like the ostrich, the rhea, and the emu, all share a common ancestor, but man, they broke off about 250 million years ago. So that's an old species in and of itself, and each has undergone its own selection pressures in the lands that it lives. They both live at the same roughly latitude, right? They couldn't make it above that equator. But where you find them is fun, right? Yay. But where you don't find them is just as telling. Here are the Polynesian, Melanesia, Micronesia, all of the Polynesian island chains. Australia has its marsupials. We have plenty of placental land mammals that go this way. And of course, we have an ocean of fish. There are bird species that exist on land. There are some reptile species that exist. And we can also explain that over here. I think the Galapagos Islands are right about there. And so you have, like, again, you have your mammals over here. You have your birds. What we find on what they call the continents and then the continental islands. So British Columbia is a continental island. Australia is like the biggest continental island we can think of. It sits by itself, but it's really close. Japan is a continental island. Papua New Guinea. When you start looking at Samoa and Easter Island and the Cook Islands, right? And you look at what isn't there in spite of having the perfect habitat that Australia has, that South America has, it makes a very good argument for evolution. And let me explain why. On big continental islands, you have a lot of niches, you have a lot of energy, you have a lot of variation. It lets animals explore and branch out their phylogenetic tree. When you get to an island is an outcrop and it's a complete isolation in the middle of the ocean. So first of all, you shouldn't have anything there to start with, but things that float in the air, pollen, seeds. There are, it doesn't sound like it. There are seeds floating around in the atmosphere as we speak right now. They're being blown over from China. And they make their way around. And soon as our season kicks up, we're going to kick up little tiny seeds into the atmosphere and they'll go hundreds and thousands of miles. Great. So once these islands start to undergo a little bit of succession and the lava that made them or whatever made them starts to undergo erosion, and you get a little bit of mineral, enough to let little lichen and moss grow, and those lichen and moss live and die, live and die, live and die, and they start to create a little bit of layer of soil that makes it good for plants. Little tiny seeds will grow. They will live and die, live and die, live and die, live and die, and they'll start to create a little bit of biota, and that biota-rich soil can give way to different species of plants, and then the plants grow and the plants grow, and then insects fly, birds fly. I'm going to stay away from bats for right now, but maybe bats fly, right? These things that can move through this medium of air can make their way around a little bit until they find a good habitat, get blown off course. And that's, you do fine, right? On islands, they're noted for birds. If you're at sea, you see a bird, you at least know you're near land. But what's missing from these islands are significant mammals. Unless we landed there with our boat and dropped off rats or dropped off pigs, sheep, or whatever, they're completely absent. If they have freshwater swimming pools, they're void of freshwater fish. They're void of amphibians. How? It's a perfect habitat for them to live in. So if you want to go to the other side and say that life was just created, boom, and this is where it existed, why would you leave such rich places for this life to exist where it was put over here, but now it's not over here? That is really the focus, I think, chapter 5 of Jerry Coyne's book, The Evidence of Land, of Biogeography. He spends a lot of time, and I put a little piece in here about it, a lot of time examining why you find species some places and you don't find them others. I'm just going to read, close your eyes. Here we go. I don't have my glasses on, so I made it 100-point font. To sum up, oceanic islands have features that distinguish them from either continents or continental islands. Oceanic islands, like the Cook Islands, Samoa, Easter Island, the Micronesia, Polynesia. Oceanic islands have unbalanced biota. They are missing major groups of organisms, and the same ones are missing on different islands. But the types of organisms that are there often comprise many similar species of radiation. And they are the types of species like birds, insects, that can disperse most easily over long stretches of ocean. And the species most similar to the inhabiting oceanic islands are usually found on the nearest mainland, even though their habitats are different. Galapagos Island. There are birds like finches that live here that are the common ancestor of the Galapagos finch. There are land tortoises that are the ancestors of the Galapagos tortoise, like the hood tortoise and the I forgot the Saint something tortoise. And there is a species of reptile, it's coming to me right now, iguanas, that's the ancestor, shares an ancestor with the marine iguana. Marine iguanas are only found on the Galapagos Islands. It's not unheard of for rafts to break off during hurricanes and make their way out to sea. Many of them perish. You can throw a basketball blindly 100 times and you're going to make a basket sometime. Maybe 1,000 times. It might take 1,000,000 times, but you're going to make one. The point being that those species, birds can fly there. Tortoises are reptiles. Iguanas are reptiles. They can go a really long time without food and eating. In fact, tortoises, when the cooks and the first settlers were going past the Galapagos Islands, that was weird. We're going past the Galapagos Islands. They would take the tortoises and they would stack them like cargo alive in the bottom of their hulls. And when they got hungry, it would be like pull a tortoise off the shelf, chop its head off, and cook it and eat it. They kept alive really well at sea. Great traveling food, like the Cliff Bars of the 1700s. But also, they can float, and if they had the advantage of floating on a raft, they could make it. Those are all the animals found on the Galapagos Islands. There's no mammals. Mammals can't make that trek. We have too high a metabolism. We need fresh water at a constant rate. Couldn't make that trek. Birds can. Seals are mammals, but I think you would figure that one out for yourself. So it makes sense that these species, where you find them for biogeography, are found there for a very specific reason. There's the idea of the tortoise. So I'm just going to wrap up with a couple more paragraphs. We can now make one final prediction. Very old continental islands, which are separated from mainland eons ago, should show evolutionary patterns that fall between those of young continental islands, like the ones we just discussed, and young continental islands and oceanic islands, like the ones we just discussed. Old continental islands, like Madagascar, right off the coast of Africa, and New Zealand, cut off from their continents 160 million years ago, and 85 million years ago, respectively, will have been isolated before many groups like primates and modern plants had evolved. Once these islands parted from the mainland, some of their ecological niches remained unfilled. This opens the door for some later evolving species to successfully colonize and establish themselves. We can predict, then, that these old continental islands should have a somewhat unbalanced flora and fauna, showing some of the biogeographic peculiarities of true oceanic islands, and they do. Indeed, that is just what we find. Madagascar is famous for its unusual flora and fauna, including many native plants and, of course, its unique lemurs, the most primitive of all the primates, whose ancestors, after arriving at Madagascar some 6 million years ago, radiated into more than 75 endemic species, meaning that's the only place you find them. New Zealand, too, New Zealand, too, has many native and most well-known being flightless birds, the giant moa, a 13-foot monster hunted to extinction about 1,500 years ago, and the kiwi, and that fat ground-dwelling parrot, the kakapo. New Zealand also shows some of the imbalance of oceanic islands. It has only a few endemic reptiles, only one species of amphibian, and two native animals, both bats, though a small fossil of mammal was recently found. It too has irradiation. There were 11 species of moas, all gone. And like oceanic islands, the species of Madagascar and New Zealand are related to those found on the nearest mainland, Africa and Australia, respectively. So you see, new islands, when they crop up, they don't get the standard whatever you have everywhere. They have to go through their own little evolutionary path too. They have to get the plants like a succession, but they're restricted because land mammals can't get there. Amphibians, of course, they would dehydrate just in the first day, can't get there. Freshwater fish can't get there. So the absence of evidence is sort of an evidence of how's that go? Absence of evidence of absence. It's a proof in a way that evolution is happening, because if it were just blanketly happening everywhere and anywhere, species popping up, then they should be popping up everywhere and anywhere, but they require very specific niches to do so. So how do I do on the proficiency scales? Well, I'm hoping you already know endosymbiont theory. We don't have to go back over that. And if you do, you should. It's a big one. I've tackled all the other ones. I think I've done a pretty good job in the proficiency scale, and I'm going to leave the Darwinian level of mastery to you. Can you evaluate evidence that I give you now? Can you look and see the strength? Of course, DNA is going to be obviously the very creme de la creme of evidence. But what about the fossil record that you have access to? What about homologous structures that you have access to? I don't know. It's up to you. It's up to you to prove to me the evidence has weight or not, given the scenario that you have. Okay? So, hope you took some good notes. This was sort of a long one, but it was all in one. Review your notes. Did you write down any questions? Do you have any questions? Email me a question. I love questions. Questions tell me that you're thinking about what you're learning. Whether you make them for me or not, if you're questioning what you're learning, you're thinking about what you're learning. That's active learning. That makes it stick. All right, friends. Hope you learned something. I'll see you guys on Tuesday. Have a great day.